so CDNE, with alpha P32 radio-labeled NTPs, and then to stop that reaction by incubating with a phosphatase that removes all of the unreacted NTPs and leaves us simply with the cyclic dinucleotides that we can image by autoradiography. We separate that reaction and visualize that reaction using thin layer chromatography. And so to remind everyone how that works, we're spotting our different complex reactions along the x-axis of a TLC plate at the bottom. And when we immerse that TLC plate in solvent, now the interaction of our signaling molecules with the mobile phase tells us something about the identity of that molecule and gives us a unique migration pattern so that we can quickly read out what CDNE is synthesizing. The real data from that looks like this. So here I'm showing you what's a pretty complicated TLC, so I'm going to take you through it slowly. But here we're spotting different reactions, again along the x-axis at the bottom of the plate, and developing the TLC so that those reactions are separated as they travel vertically, and we're varying which enzyme we add to our reaction mix. So each of these lanes represents a different enzyme product, and moving from left to right, the first 4 products are our known cyclic dinucleotides, either from eukaryotes or bacteria. What I want you to appreciate is that the last lane here, which is the CDNE lane, produces a spot that's different from the 4 other spots that are produced by our known cyclic dinucleotide synthases. So the takeaway from this is that CDNE is synthesizing a novel compound that's distinct from the previously identified cyclic dinucleotides. In data I don't have time to show you, we were able to go further and ask, well, what are the constituents within this spot? So we expect it to be a cyclic dinucleotide based on the fact that it's migrating similar to these other cyclic dinucleotides, but by varying the reaction conditions and really where the alpha P32 label is, what we were able to find was ATP and UTP are necessary and sufficient for product formation. That allowed us to finally determine that the product of CDNE wasn't CGAM, but was in fact cyclic UMP-AMP. Cyclic UMP-AMP is the first pyrimidine-containing cyclic dinucleotide, and this really was surprising to me because when you go to the nucleotide signaling literature, in general, pyrimidines are conspicuously absent. So we've all learned about the cyclic mononucleotides like cyclic AMP and cyclic GMP, but in general, we don't find pyrimidines being incorporated. And so in order to understand exactly how this was happening at a molecular level, we took a structural approach. In collaboration with a graduate student in Philip Cranch's lab, James Eaglesham, we were able to determine the crystal structure of CDNE to 1.1 angstrom resolution. And when we did that, what we found was that indeed CDNE was CGAS-like, similar to DNCV, not sharing any sequence similarity, but structurally sharing this pol beta-like nucleotidyltransferase pol. And so in order to appreciate that, I hope you can align by eye the beta sheets here in blue that are in the active site and see that the core of DNCV, CGAS, and CDNE are all highly similar. For this reason, CDNE was named for CGAS or DNCV-like nucleotidyltransferase from E. coli. But the other thing that we were able to do, armed with this structural information, was begin to define exactly what we were describing in these bacterial CGAS-like enzymes. Having 3 different enzymes that were all highly similar, we decided to name this family of related proteins CGAS or DNCV-like nucleotidyltransferases. And I'm going to use this abbreviation of CDNTases going forward because what I next wanted to do in this project was to continue on in that question of, well, are there other bacterial CGAS-like enzymes? And by proxy, are those bacterial CGAS-like enzymes also synthesizing molecules that have an important role in immune signaling? In the beginning, we started with that unsophisticated approach, but by having this structural information, what we were now able to do was to construct a discovery pipeline that really relied on the structural features of these genes in order to collect up as many CGAS-like enzymes predicted to be encoded by bacteria in the publicly available databases. When we did that, and I should point out that we were largely enabled by this publication here at the bottom from Maxwell Burrows in the Arvin group at the NIH, what we were able to determine were 5,600 bacterial C-gas-like enzymes, or CDNTases, in the NIH databases. Those CDNTases are found very broadly distributed among bacteria. So that 5,600 actually describes over 16,000 bacterial genomes that are encoding these C-gas-like enzymes. And that's because many of these enzymes are encoded identically in different genomes. That's how we get a different number of genomes from unique sequences. 16,000 bacterial genomes represents over 10% of the sequenced bacteria in the database. And so our assertion from that is that over 10% of bacteria encode C-gas-like enzymes. It's a little easier to point out all the bacteria that are not found to encode CDNTases than it is actually to talk about all the phyla that are represented. But I will point out that bacteria with reduced genomes or obligate intracellular organisms are conspicuously absent from this list. The other takeaway from these bacterial C-gas-like enzymes is that they're almost exclusively encoded on mobile genetic elements. And I can't tell you exactly the significance of that right now, but I think it's telling us something about the biology of these genes, and in the future, I'm hoping to be able to understand exactly why that might be. The goal of understanding these CDNTases, as I pointed out, was really to be able to understand what is the biochemical landscape of molecules that are available to the host from bacteria in those interactions. But we can't possibly look at 5,600 different sequences or different proteins, and so instead we decided to define related clades of these CDNTases so that we could look at type enzymes and really particular candidates from each clade in order to make broader generalizations. The way we did that was to align all those 5,600 sequences and begin to group the proteins based on their relatedness to one another. And I'll just pause here because this is the raw tree that we were able to construct by aligning all these proteins. But what I want to point out is really the incredible diversity among these sequences. So if you can see down at the bottom left, our legend represents about 0.5 substitutions per site. And that means that this distance, if you look at, say, sequences on one side of the tree versus the other, represents over 1 substitution per site. So virtually no amino acid identity is shared across this tree, but all of them are unified as being predicted to be C-gas-like in structure. What I did next was to group each of these proteins together based on their amino acid identity. And so here the colored different clades represent about 25% identity. And the goal here is, as I was pointing out, to be able to make broader generalizations about the phylogeny of these enzymes. So DNCB is found in this clade A, and CDNE, the enzyme that I was talking about earlier, is found here up in clade E. And so you can see that there are many other examples from across the tree that we were hoping to sample. And the way we went about doing that was to clone out 66 representatives from across the tree. We chose those representatives based on the genus and species of the bacteria that encoded each of these CDNTases, and we biased this analysis towards bacteria that are predicted to interact with eukaryotes. And again, that was because we're really excited about finding molecules that have some sort of an immune stimulatory capability. Each of these red dots represent one of the genes that we were able to synthesize, clone into a bacterial expression vector, and then return back to our TLC assay to simply screen which of these are producing products, which are producing new products, and then finally assign each of these different clades a specificity. So to remind you what that assay looked like, here we're incubating each of our enzymes with alpha P32 radiolabeled NTPs, so all 4 NTPs. We're assaying for a signaling molecule, and in the left, here is a DNCB reaction. In the first lane and in the second lane, we have a catalytically dead version of that protein being added. So here you can see where 3' CGAMP runs. That's what this arrow is denoting. And in the next lane, you can see there's relatively low background in this assay. We could then expand our analysis to now look at here, this is the first 20 different CDNTases that we looked at. And we could find a few important features